Hello everybody, my name is Sandra Gonzalez. I am a postdoctoral researcher at the University of Yanana at the Department of Biological Applications and Technology. First, I would like to thank the organizers of the first Yanina Science Festival for inviting me to participate in this event by giving a short talk. And today I'm going to talk to you about some aspects of human evolution, more specifically our face evolution. My expertise is in developmental biology, and I study a type of cells which appear really early during embryo development, and they are in charge of shaping our face. So the story I'm going to tell you about today is how these cells might have been implicated in shaping our face throughout evolution. So I hope that this story will interest you enough so you can go and deepen your knowledge in human evolution and development. We see ourselves in the mirror every day. Sometimes we may take our face for granted, but next time you do, please spend like 1 minute or two thinking of the millions of years that have gone to shape your face. So after all this time, you are here today, and our faces carry an evolutionary history which we should be proud about. It is common knowledge, or at least most people have heard, that humans and our closest relatives alive today, which are the chimp and the bonobos here, we share about 99% of our genetic material. If we go back in time and we look at the amazing evolutionary history that went before we appeared, it seems that about 7 million years ago, we split from the chimpanzee's lineage. And some people suggest it may have been a bit later, about 5 million years ago, but sometime around that period, the first hominin emerged. Hominins are direct lineage of the human today. All the hominins found on the fossil record are now extinct, but they have given us clues to the step-by-step reshaping of the facial features, leading to what we know as a human face today. Many people may ask why. Why did we come about? Why did humans appear on Earth? This is quite a philosophical question, and it is probably impossible to answer. However, the how begins to get some answers. Evolution and the history of life on Earth is fascinating. Species have evolved and adapted to their environment and successfully survived and reproduced. All living species that we know today can be said to be the fittest of all those that have existed before. Now, this does not mean that they are stronger, but that they are the best adapted. Adaptation comes with a beneficial trait. So each species have evolved their own to thrive in their own environments, but vertebrates like us exclusively invented a type of cells called the neural crest. So the neural crest is an outstanding, remarkable cell type that arises very early during embryo development. In humans, it appears about 3 to 4 weeks of gestation. So you can imagine that the woman may not know yet that she is pregnant, but this is a very, very crucial time for proper development. Now, the neural crest in humans is also very difficult to study. So scientists have chosen other model organisms or other animals in order to study the function and their behavior. So here I'm showing you, here at the corner is a schematic of a chicken embryo. This is how it looks after a few days of fertilization. And scientists have developed a technique that can label any type of tissue with fluorescent markers. This helps them a lot to visualize the tissue of interest, in this case, the neural crest. And as you can see here in green, you can see a huge patch of cells that are coming out of the midline. The midline is what it will become, the central nervous system. And that's why they are called the neural crest. However, these cells don't only make neurons. They also make cartilage, bones, smooth muscle, and in the trunk at the bottom here, like what I'm showing here is the head, but at the bottom you see the trunk neural crest. In the trunk neural crest, they give rise to the pigment cells and some neurons. The pigment cells are very important because they protect you under the sunshine and give you the glowing aspect of after your summer holidays. Now, all vertebrates have trunk neural crest, but this guy here, the lamprey, does not have a jaw, neither a skull, compared to the one on the right that has these amazing jaws that have helped him to become the #1 predator in his habitat. So the cells that gave him this advantage are, of course, the neural crest. But these are a specific kind of neural crest called the cranial neural crest. To study in more detail the cranial neural crest, scientists have used widely the frog embryo. This little ball here is a frog embryo. And here it's his head. Again, in green, you will see the cranial neural crest labeled with the fluorescent green marker. And so they can track and see where it goes. You can see that it's moving away from the top, migrating down, and populating what it will become the front part of the face. So here is a schematic of what happens in the human embryo. So at the top, you see a schematic of about 4 weeks of gestation of a human. And in green, you see the neural crest tissue. And eventually, you see how in the adult that developed the facial bones. So we can conclude here, and this image here tells us that the neural crest that moves away and populates the front parts of our face will become the bone and the facial bones. So they are really responsible of the shape of our face. Now that you know a little bit more about the cells that are behind shaping our face, let's go back to the fossil record. So scientists have been fascinated, and many people as well, fascinated with the human evolutionary history. And in the fossil record, there are stepwise samples that lead to the human face as we know it. So do you notice any pattern in this picture here? Can you notice that there's a polished aspect in our species, like in the Homo sapiens? It's like retracted. It has this retracted aspect that makes us look friendly. These are our cousins, the chimps. On the left, you see a calm and somehow friendly pose. But it's still, if we see it in front of us, it's kind of intimidating. It has really wide jaws, and when it's not happy, it looks quite scary. As you can see, humans have the same expressions, but we look much friendlier, even if we are angry. So can you think of any other example that shows related species and one of them looks friendlier than the other one looks more savage? One example is the house cat compared to the tiger. So these are more or less comparable to us and the chimps. They share about 95% of their genetic code, and they had a common ancestor about 10 million years ago. So what do we all have in common? What do we have in common with the house cats? The domesticated cat looks friendlier, even if it doesn't stand out as the most sociable animal, domesticated animal. It is a lot more sociable than the tiger, and it is also more cooperative and calmer. We called it the Domestication Syndrome. So I'm not going to deepen too much on this, but generally it refers to a theory suggesting that humans underwent a process of self-domestication, in which we preferred a calmer and more cooperative partner. That eventually helped us to start living in groups. And this process was closely related, or at least it was thought to be closely related to our facial structure and our facial gestures, that in the way that the most friendlier would be more successful in finding a partner. This would lead the humans to slowly get the appearance that we know of today. This theory just found a possible molecular explanation. So scientists from Spain, Germany, and Italy worked together and discovered a genetic link to reshaping the face. And guess what? Yes, the neural crest might be a major player on this event. They studied two syndromes. These are interesting because they present opposing characteristic facial conditions linked to behavioral and cognitive abilities. So they are called Williams-Burton syndrome and the 7q11.23 duplication syndrome. So they probably won't say much with their name, but I'm going to try to explain a little bit Williams-Burton syndrome so you can understand why they chose these particular conditions for their studies. It is peculiar that the same region in the genetic code is affected in both syndromes. Here I'm showing you that 1.5 million base pairs are affected in chromosome 7. Here it's a schematic representation of the chromosome. And in Williams syndrome, there is only half dose of this region here, whereas in the duplication syndrome, it has one extra copy of this region. X-ray photographs tells us a lot about our bones. And here there are X-ray photographs of a normal child skull on the left, and on the right it is a skull of a 5-year-old kid with Williams-Burton syndrome, which carries only half of the gene dose. You can see clearly that the face is retracted compared to a normal kid's skull. The face has characteristic wide nose, full lips, and very short mandibles. Along with a retracted face, it is very characteristic of these kids and people in general that have this syndrome, that they are extraordinarily friendly and they are incredibly social characters. They also have an acute sense of hearing. Their listening is amazing, which makes them outstanding in music, like this guy here, Alex Sweezy. What is interesting is that in the duplication syndrome shows completely opposite characteristics to the Williams-Burton syndrome. I cannot show you pictures, but the literature explains that they are shy, aggressive, and their cranial aspect in some occasions show a bigger size than the normal. So this suggests that this region in the chromosomes contains interesting genes involved in shaping the face and influencing behavior. Two things that were pretty important in human evolution. Using innovative techniques, the scientists found that a gene called BAZ1B is a master regulator of about 40% of the genes involved in neural crest development. They found that depending on the dose of this gene, the cells, the neural crest cells, reduced in number and migrated less as well. So they showed that delay in arriving to the regions that are going to become the face later on. So, as you can imagine, the less the gene, the less prominent your face will be. For the relevance of this study, they went back to analyze the DNA of archaic humans. You can see here the prominent eyebrow bone and the protruding maxilla, which are characteristic of this species. So what these scientists did was to compare the same region in chromosome 7, the one that is affected in Williams-Burton syndrome, between the archaic and the domesticated human DNA. What they found was pretty amazing. The modern human carries a number of mutations in this region, including the gene BAZ1B, which we said before, suggesting that our present face shape is the result of a mild neural crest deficit, which defined our softer characteristics. Along with that, it may also have affected our cognitive abilities and basically giving humans the advantage of adapting and creating societies that develop the tricks to survive in different environments. Well, here I finish my talk. Thank you again to the organizers for inviting me to participate. I presented the fascinating work of Mateo Zanella and colleagues. Thanks everyone for listening, and I hope you enjoyed the rest of the Science Festival.